Thanks for joining us on another exciting journey into the world of science, right here on the SANS Science Experience. If you enjoyed this kind of content, hit like and subscribe. It's the best way to support the channel and keep these episodes coming. Today, we're plunging into the incredible world of mitochondria, those tiny compartments within our cells. You probably know they're the powerhouses, generating energy. But our sources reveal they are so much more than just ATP factories.
Absolutely. They're fundamental metabolic hubs. They orchestrate a surprisingly vast array of processes, way beyond just, energy production. And consider this, they even possess their own ancient bacterial origins. It's quite amazing, really, a testament to how critical they are.
It's mind-boggling to think about that evolutionary history. Okay, let's delve a bit deeper then. Something that really jumped out from our research is just how dynamic these mitochondria are. They aren't static things at all, are they?
Yeah. No, not at all. That's a really crucial insight. Their internal makeup, the specific proteins they contain, it's in constant flux. It adapts to what the cell needs right then, its developmental stage, even outside cues. It's an incredibly adaptable system. We can think of it as mitochondrial plasticity.
All right, plasticity. And this brings us neatly to the heart of our discussion today. Mitochondrial proteases, or mitoproteases. Now, my initial thought, maybe like a lot of people, was that these were primarily involved in, cleaning up cellular mess. But it sounds like there's a much more intricate story.
That's a common starting point, yeah. The early focus was definitely on their quality control job finding and degrading proteins that are misfolded or damaged. They act as a sort of primary defense mechanism, keeping the mitochondria healthy. But the really exciting discoveries more recently highlight their vital regulatory roles. They're actively shaping and directing loads of mitochondrial processes.
Okay, so the goal of this deep dive then is to really unpack these multifaceted roles. We want to look at both the essential quality control stuff and these emerging regulatory functions, but why should you, listening at home, care about these tiny protein chopping enzymes?
Well, because these seemingly small molecular machines have a huge influence on our overall health, the aging process, and the development of a wide spectrum of diseases. By getting a deeper understanding of how they work, we can unlock really valuable knowledge about these fundamental biological processes. And we've gathered quite a range of studies to help us explore this.
All right, let's get down to brass tacks. What exactly are these mitoproteases?
In essence, they are proteases, so enzymes that break down other proteins, and they live inside the different compartments of the mitochondria. And there are a lot of them. We know of at least 45 distinct ones, and 23 of those seem to be exclusively mitochondrial. They don't function anywhere else.
45. That's quite a crowd in such a small organelle. So what are their main jobs, broadly speaking? What are the overarching functions?
They do several critical things. One key function is preventing the buildup of short-lived regulatory proteins, making sure these signals don't stick around too long. They also play a crucial role in modulating the activity of other mitochondrial proteins through processing, basically fine-tuning function by snipping them at specific spots. And of course, the big one, degrading damaged proteins. That's fundamental for maintaining proteostasis, that balance between making and breaking down proteins.
So it's like they're the sanitation crew, the maintenance engineers, and maybe even the conductors of the mitochondrial orchestra, all rolled into one. That gives you a real sense of how vital they must be, right?
That's a great way to put it, yeah. And just to quickly categorize them, we have processing peptidases. They mainly snip bits off proteins. Then there are ATP-dependent proteases using energy to unfold and degrade targets. Oligopeptidases handle small peptides. And then there's a kind of catch-all category of other mitochondrial proteases doing various jobs. We can look at Table 1 in the sources later for specifics if needed.
Okay. And you mentioned something called pseudomitoproteases. That sounds contradictory.
Yeah, they're fascinating, actually. Structurally, they look like proteases, but they're missing the key bits needed to actually cut proteins. But they're not useless. They often play important roles, like being structural parts of the larger protease complexes.
Right, okay. So we have a good handle on what they are. Now let's really dig into that central idea, the shift from just seeing them as quality control to recognizing their regulatory power. You touched on this.
Yes. Historically, the main view was quality control, identifying and getting rid of misfolded or damaged proteins. They were seen as the first line of defense against proteotoxic stress, acting before the cell might need to do something drastic like mitophagy, right, degrading the whole mitochondrion.
But the more we look, the clearer it becomes that's only half the story. What really drove this change in thinking?
Well, just a growing body of research has increasingly shown their active, essential roles in regulating a whole spectrum of mitochondrial processes. It's not just tidying up, they're actively controlling things.
So how do we differentiate between these quality control substrates, and regulatory substrates. It can't always be obvious why a protein is being targeted, surely.
That's a good question. Quality control substrates are generally targeted because they're damaged, or they haven't folded right, or they haven't assembled correctly into bigger structures. Think of them as faulty parts getting chucked out. Regulatory substrates, though, are often degraded really quickly even when everything's fine. Their levels are kept deliberately low to allow for rapid shifts in cellular processes. If you inhibit the mitoprotease that normally degrades a regulatory substrate, that substrate piles up. That's a key clue.
You gave examples like EMRE and the prelitips family. What's so intriguing there is how they're specifically targeted for such rapid turnover in healthy mitochondria.
Yeah.
It's not like they're damaged, right?
Exactly. That specific targeting is a huge area of research right now. How did these proteases pick out just those regulatory proteins at just the right time? The exact mechanisms governing that selectivity. Still largely a puzzle.
Okay, let's transition then. How do these mitoproteases actively control key mitochondrial processes? Protein import and getting proteins to the right place seems like a fundamental area where they'd be critical.
Absolutely. One of the most well-established roles is in maturing newly made proteins heading for the mitochondria. Most are made outside, encoded in the nucleus, right? Many carry this N-terminal signal, the mitochondrial targeting sequence, or MTS. MCP, that's the mitochondrial processing peptidase, snips off this MTS once the protein arrives in the matrix. It's a crucial step for the protein to fold correctly and find its spot. And after that initial cut, another protease, PrepP, can degrade the leftover MTS fragment.
So like removing the shipping label once the package arrives.
Precisely. And if that first processing step goes wrong, it can cause serious problems. We know, for instance, defects in processing proteins like frataxin, potentially involving YME1L, are linked to certain diseases.
But it's not always just about removing that initial signal. Sometimes more trimming is needed for stability or function.
Yes, exactly. Enzymes like MIP, METUP1D, and XNPEP3 can do further trimming. This can boost the stability of specific proteins. A good example is MSRPL12, part of the mitochondrial ribosome. It gets processed sequentially by both MPP and MIP. And this actually indirectly affects mitochondrial transcription by making sure the RNA polymerase complex is stable.
Interesting. And then there's targeting to the intermembrane space, the IMS, that bit between the inner and outer membranes. That often involves a two-step process, I think.
That's right. Proteins heading for the IMS often have a bipartite presequence, a two-part signal. First, MPP makes a cut in the matrix. Then IMMP, the intermembrane peptidase, makes a second cut, releasing the mature protein into the IMS.
IMMP sounds quite versatile. You mentioned it's pleiotropic.
It really is. It can cut substrates at both ends, N-terminus and C-terminus. And it even helps sort proteins that are actually meant for the outer membrane eventually. Peroxyredoxin, an antioxidant enzyme, is a great example. IMMP cleavage sends it to the IMS, but a different pathway involving MPP and then MIP sends it to the matrix. Different processing, different destinations.
What about the import machinery itself? It's not just the proteins coming in. The channels must be regulated too, surely.
Absolutely. YME1L, that IMEA protease in the inner membrane, can actually degrade certain subunits of the TM23 complex, which is a major import channel. This can help manage the protein load, especially under stress. And interestingly, YME1L also affects TM23's specificity by degrading ROMO1, a regulatory bit of the channel. So YME1L acts like a gatekeeper, tweaking not just how much gets in, but also what gets in preferentially.
And then there's this intriguing thing about dual localization. One protein doing jobs in mitochondria and somewhere else in the cell. Do mitoproteases play a role there too?
They certainly can. Take fumarase, a key enzyme in the TCA cycle in mitochondria. It also works on DNA repair in the nucleus. How efficiently it gets imported can be limited by how it folds outside, and MPP processing during import can actually cause some of it to get kicked back out into the cytosol, so the processing itself influences where it ends up.
That's a clever way to manage multitasking proteins. And PRL seems to have a particularly unique way of affecting localization.
Yes, PRL is pretty special. It can cut proteins within their transmembrane domains, those bits that anchor them in the membrane. This lets PRL effectively release proteins from the inner mitochondrial membrane, changing where they are located. START7, involved in lipid transfer, is a key example. PRL cleavage affects its balance between the IMS and the cytosol. This intramembrane cutting is also vital for proteins like PGAM5 and PNK1, which are crucial for mitophagy clearing out damaged mitochondria. Normally, PRL cuts PNK1, leading to its degradation outside, which prevents mitophagy happening unnecessarily. But when mitochondria are damaged, this cutting stops, PINK1 builds up on the outer membrane and signals time for removal. It's worth noting, though, that other proteases like MPP, CLPXP, and AFG3L2 are also involved in getting rid of PINK1, though we're still figuring out exactly how.
Wow. It really is like a meticulously choreographed dance of proteases, deciding where proteins go and what happens to them.
Okay, let's switch gears slightly. What about maintaining the mitochondrial genome and expressing its genes? Another critical area where mitoproteases seem deeply involved. Absolutely vital. You need tight coordination between the nuclear genome and the mitochondrial one for the oxidative phosphorylation system, the main energy generator, to work properly. Most mitochondrial proteins come from nuclear genes, but the mitochondrial DNA, mtDNA, encodes some crucial components itself. Mitoproteases are key players in keeping mtDNA stable and regulating its gene expression.
TFAM, mitochondrial transcription factor A, sounds like a really important one here.
It is, absolutely. TFAM is essential for both copying mtDNA and packaging it up correctly. Too little TFAM, you lose mtDNA. The right amount is crucial for stability and for transcription. LONP1, a protease in the matrix, controls TFAM levels. It degrades TFAM when it's not actually bound to mtDNA. And interestingly, phosphorylation of TFAM can make it more likely to be degraded by LONP1, another layer of control.
So LONP1 is like a guardian, making sure there's just the right amount of TFAM around. And it seems LMNP1 might even interact directly with the mtDNA itself.
The evidence points that way, yes. LONP1 seems to like single-stranded DNA and G-rich regions in mtDNA. This interaction could potentially influence both starting replication and starting transcription. It's got multiple roles in safeguarding the mitochondrial genome.
What about handling the messenger RNA, the mRNA made from mtDNA, and the actual protein translation inside mitochondria? Mitoproteases involved there too.
Yes, they are. LRP-PRC, a protein involved in processing mitochondrial mRNAs, is a known target of the CLPXP protease. And if you look at building the mitochondrial ribosomes and translating proteins, the m-AAA protease complex is critical for processing misRPL32, a ribosomal subunit. That processing step is essential for the ribosome to work. There might even be a neat negative feedback loop. If damaged proteins build up in the matrix, they might compete with misRPL32 for the m-AAA protease. That could act as a signal to slow down mitochondrial protein synthesis.
That's clever, linking protein quality control directly back to the protein production machinery. And CLPP, another protease, also seems key for ribosome assembly.
It is. CLPP degrades ERL1. ERL1 is needed early on to assemble a ribosomal subunit, but it has to be removed for the final working ribosome to form. If CLPP isn't working properly, ERL1 builds up, mitochondrial translation is impaired, and that's linked to some severe health issues.
So mitoproteases aren't just cleaning up afterwards. They're actively involved in building the protein-making factories themselves. Okay, let's move to another crucial area, assembling proteins within the mitochondrial membranes.
Right. This is where mitoproteases ensure those big protein complexes in the intermembrane, like the electron transport chain components, get put together correctly and function efficiently. They can do this by precisely processing specific subunits or by controlling the turnover, the lifespan of others.
The ATP synthase, that amazing molecular motor that makes ATP, is one example. A specific protease, ATP-23, is involved there.
Exactly. AT23 in the IMS helps mature ATP6, an mtDNA-encoded subunit. It also acts like a chaperone, guiding its assembly into the final ATP synthesis complex. And PARL, that intramembrane cutter, processes TTC-19, which is vital for complex 3 of the electron transport chain. Losing PRL function leads to complex 3 deficiency and severe neurological problems.
And it's not just about the initial build. Mitoproteases also help these complexes adapt to changing conditions, like low oxygen.
That's a fascinating point, yes. LONP1 is crucial for letting cytochrome C oxidase complex 4 adapt to hypoxia. It degrades one subunit, COX41, which then allows a different subunit, COX42, to slot in. COX42 works better when oxygen is scarce. It's a remarkable example of proteolysis driving adaptation.
Incredible fine-tuning. And then there's the MCU complex, the calcium channel, essential for controlling calcium levels in mitochondria, another example of regulation.
Absolutely. The m-AAA protease complex degrades excess EMRE, a key helper component of the MCU. This ensures the channel only forms functional units when it's properly regulated by MICU1 and MICU2. It prevents uncontrolled calcium influx and potential overload, which can be lethal for the cell.
It really seems like mitoproteases are constantly adjusting and maintaining the balance of these essential machines, and even the proteases themselves are controlled this way.
Yes, indeed. The MAA subunit AFG3L2 helps assemble itself and is needed to mature another MAA subunit, SPG7. It's a nice self-regulating system. Similarly, YMA1L limits the levels of ROMA1, influencing the import machinery's specificity and even regulating its own import.
This intricate web of control is just astounding. Let's shift now to something I have found particularly interesting, the link between mitoproteases and mitochondrial phospholipid metabolism. It's not just about the proteins, is it?
Not at all. Mitochondria, especially their intermembrane, are packed with lipids. These are absolutely crucial for structure and for the function of all the embedded proteins. And we now know mitoproteases are important regulators of the mitochondrial lipidome, the whole collection of lipids there.
You mentioned PE, phosphatidylethynolamine, and cardiolipin as key lipids. And their synthesis needs precursors imported from the ER, the endoplasmic reticulum. How do mitoproteases fit in?
Well, transporting those precursors, like phosphatidylserine, PS, and phosphatidic acid, PA, across the IMS relies on specific lipid transfer proteins. The i-AAA protease YME1L has been shown to degrade several of these transfer proteins, including the prelitups family and STARD7. By controlling their levels, YME1L directly influences the phospholipid makeup of the inner membrane.
So they're regulating the supply chain for these essential lipids. And it seems to be a two-way street. The lipids themselves can affect protease activity.
Exactly. For instance, lower levels of PE in the inner membrane can actually speed up YME1L's protein cutting activity. It shows this tight reciprocal link between protein and lipid balance. Plus, mutations affecting PE synthesis can also mess with the activity of certain mitoproteases.
And LACTB, another IMS protease, seems to have a role in regulating PIS, an enzyme making PE, particularly in cancer cells.
Yes, LACTB can lower PISD levels in cancer cells. This affects PE levels and might influence how these cancer cells grow and survive. It really highlights how mitoproteases can impact broader cell processes, not just core mitochondrial stuff.
Okay, let's tackle mitochondrial dynamics next. Continuous fusion and fission, the merging and splitting. It's visually striking and seems vital. How do mitoproteases manage this?
This dynamic balance between fusion and fission is absolutely crucial. It maintains mitochondrial health and lets them respond to the cell's changing needs. OP1, a protein in the inner membrane, is key for fusion and also for maintaining the cristae structure, those inner membrane folds. And OP1 activity is tightly regulated by mitoproteases.
OME1 and YME1L are the main ones processing OP1, right? Turning the long LOP1 form into shorter SOP1 forms.
Precisely. The ratio of LOP1 to SOP1 dictates the shape and connectedness of the mitochondrial network. Generally, LOPA1 promotes fusion. The balance between L and S forms, controlled by OMA1 and YME1L working somewhat in opposition, maintains a healthy tube-like network.
And it sounds like OME1 responds more to stress signals, while YME1L is perhaps more tuned to the cell's metabolic state.
That's the general picture emerging, yes. Stress conditions like losing membrane potential or problems with energy production can activate OMA1. This leads to more OP1 processing and consequently more mitochondrial fragmentation. YME1L, on the other hand, seems more responsive to metabolic signals. It can even be activated by low oxygen or starvation, leading to OP1 turnover.
And it's not just a one-way street. The activity of these processing enzymes themselves is also regulated.
Yes, indeed. OMA1 can actually degrade itself autocatalysis once it's activated. The stability of both OMA1 and YME1L can also be influenced by ATP levels during stress. YMA1L can also undergo autocatalysis or even be degraded by OMA1 under certain conditions. This allows the network to become more fused again once the stress is over.
So, a highly sophisticated system ensuring dynamics can respond rapidly and appropriately. And OP1 itself does more than just control shape.
Exactly. Beyond fusion, it's critical for cristae structure and for the respiratory chain's efficiency. While OMA1 and YMA1L mainly impact dynamics via OP1 processing, these other OPA1 functions seem more dependent on its overall amount, not just the LS ratio.
Now let's delve into a more somber aspect, cell death. How do mitoproteases factor into the decision for a cell to live or undergo programmed destruction, apoptosis?
Mitochondria aren't just bystanders in apoptosis, they're active players. Fragmentation often happens early on, and releasing pro-apoptotic proteins from the IMS into the cytosol is a critical trigger for the death cascade. Mitoproteases influence this both directly and indirectly.
You mentioned the SBY complex SOP2, PRL, and YME1L as being important here.
Yes, this complex in the inner membrane seems to act as a sort of proteolytic hub, regulating both dynamics and cell death pathways. YME1L can indirectly influence cytochrome C release, a key step by affecting dynamics. More directly, PARL can cleave SMAC diablo, another pro-apoptotic protein in the IMS, helping it get out into the cytosol to promote caspase activation. IMMP2L is also implicated. It can cleave AIF, apoptosis-inducing factor, which triggers cell death even without caspases.
And it seems that OP1 processing balance, OL1 versus YR1L, is again crucial for a cell's susceptibility to death.
That's right. Cells lacking YME1L often have very fragmented mitochondria and are more prone to cell death. Interestingly, if you also remove OMA1, this increased susceptibility can be suppressed. It suggests that OMA1-driven OPO1 processing and the resulting fragmentation can push cells towards death in some contexts. But it's important to remember these effects can be tissue-specific. PARL also seems important for apoptosis progression, maybe through SMAC processing and interactions with apoptosis inhibitors.
So mitoproteases are active regulators in cell death, influencing key signals and the cell's ultimate fate. Okay, let's shift to how mitochondria react to stress. Mitoproteases seem involved in sending out internal SOS signals.
Absolutely. When mitochondria face stress, like protein buildup in the matrix or IMS, various signaling pathways kick in to try and restore balance. Mitoproteases are key players in these stress responses, often leading to changes in gene expression.
The UPRmt, the mitochondrial unfolded protein response, is a key pathway. In worms, C. elegans, it seems CLPP and LNP1 are central.
Yes, in C. elegans, it's thought CLPP degrading misfolded proteins in the matrix triggers UPRmt. A transcription factor, ATFS1, normally gets imported and degraded by LONP1. Under stress, maybe import is reduced, ATFS1 goes to the nucleus instead and turns on genes for chaperones and protease to fix the problem. However, the pathway looks a bit different in mammals. There, LONP1 is the main quality control protease in the matrix, and CLPP's role in triggering UPRmt is less clear. Mammals also have a broader integrated stress response triggered by various mitochondrial issues.
What about stress originating specifically in the intermembrane space? Are there separate responses for that compartment?
Yes. Overexpressing misfolded NDOG, an enzyme, in the IMS triggers a specific response. HTRA2, an IMS protease, normally degrades it. But if it builds up, it can lead to increased ROS production reactive oxygen species. This activates downstream signaling involving protein kinase B and estrogen receptor R8, ultimately boosting protective gene expression.
And then there's the intriguing star overload response. Accumulating the star protein involved in steroid synthesis actually causes several mitoproteases themselves to be upregulated, like a feedback loop to boost quality control capacity under pressure.
Exactly. It's a beautiful illustration of these intricate networks involving mitoproteases, all working to keep mitochondria healthy under various challenges.
Okay, let's try to connect all these fascinating details back to the bigger picture. Human health and disease. We know mitochondrial dysfunction is involved in so many conditions: neurodegeneration, cancer, aging. How do mitoproteases fit in?
Well, given their incredibly diverse and vital roles, it's maybe not surprising that mutations in mitoprotease genes are linked to a wide range of human diseases. And beyond mutations, even just changes in their normal expression levels might contribute to disease development or progression.
Table 2 in our sources gives a glimpse. It's quite striking how diverse the diseases and affected tissues are.
Yes, and because they're involved in such core processes, these mitoprotease-related diseases often look similar to other primary mitochondrial diseases, often affecting multiple systems' pleiotropic effects. A key challenge is figuring out the root cause. Is it mainly damaged proteins building up due to bad quality control, or is it the deregulation of specific processes normally controlled by that protease? Probably a mix in many cases.
The example of Kodo syndrome, linked to LONP1 mutations, seems like strong evidence for protein accumulation being a major problem.
That's a clear case, yes. LONP1 mutations lead to these dense protein clumps in the matrix. Strong evidence for a primary defect in degradation. But for many other mitoprotease diseases, the evidence for widespread protein misfolding isn't as obvious. It suggests losing specific regulatory functions might be the main driver of the pathology.
Let's focus briefly on neurodegeneration. The m-AAA protease subunits, SPG7 and AFG3L2, are strongly implicated in things like hereditary spastic paraplegia and spinocerebellar ataxia.
Yes, and the specific symptoms often correlate with where these subunits are expressed in the brain and with isoenzymes form in different cell types. Mouse models lacking functional AFG3L2 show a whole range of problems: issues with transport along axons, calcium overload, weird mitochondrial shapes, problems with mitochondrial protein translation, and respiration defects. It really underscores how critical m-AAA is for neurons. Interestingly, Fixing the mitochondrial shape by also deleting OMA1 didn't fix the transport problems. But the link between AFG3L2 loss, calcium overload, and the death of specific brain cells, like Purkinje cells, seems particularly strong.
What about cancer? That seems like an area where the connection is getting more attention.
Indeed. A number of mitoproteases show altered expression in tumors compared to normal cells. This suggests they're involved in tumor development and spread. LACTB, for instance, looks like a tumor suppressor in some breast cancers. Conversely, LONP1 is often overexpressed in various tumors, linked to more aggressive behavior. Mouse studies show reducing LONP1 can actually protect against some tumors, and high LONP1 in human tumors often means a poor prognosis. There's also growing evidence that CLPP is important for the metabolic changes in cancer cells' metabolic reprogramming. This might offer a way to selectively target cancer cells. And HTRA2 seems involved in regulating tumor suppressors and preventing invasion.
It's fascinating and a bit unsettling how these enzymes meant for cell health can be dysregulated or even hijacked in cancer. Finally, let's touch on aging. Mitochondrial dysfunction is often cited as a major factor there.
Yeah, as we age, mitochondrial activity tends to decline. We often see more fragmentation, changes in cristae structure. This is linked to declining proteostasis, damaged proteins building up. Interestingly, LONP1 levels, the protease clearing damaged proteins in the matrix, tend to decrease with age in some tissues. But this can sometimes be reversed by things like caloric restriction. And in some simpler organisms, boosting LONP1 can actually promote longevity. IMMP2L is another one linked to aging. Genetic variations near its gene are associated with living exceptionally long lives. Conversely, mice lacking MP2L show signs of premature aging, but it's complex. Downregulating IMMP2L has also been linked to cellular senescence, which can be protective in some ways.
So it's not as simple as saying more or less of a specific mitoprotease is always good as we age. It's likely about maintaining a very fine balance.
Exactly. Given mitochondria's flexibility and the sheer range of mitoprotease functions, their roles in aging are bound to be complex and context-dependent, varying between tissues, different conditions. The fact that both impaired function and unbalanced expression can cause disease really highlights how critical it is to coordinate their activity properly throughout life.
This has truly been a deep dive into the multifaceted world of mitoprotease. It's really clear that just thinking of them as quality control was a major underestimation.
Absolutely. Their regulatory roles are now seen as central to mitochondrial biology, and the diseases linked to them are complex, often overlapping with other mitochondrial disorders. Understanding their dual roles, quality control and regulation is essential, especially if we want to unravel the complexities of aging.
And there are still so many basic questions left, aren't there? How do they pick out the right substrates with such amazing specificity? How is their own activity controlled by the cell's changing needs?
Exactly. And how does their location matter? These potential proteolytic hubs in the inner membrane and their relationship with the surrounding lipids, that whole environment, these are all exciting, active research areas. Future discoveries are bound to surprise us and deepen our appreciation for these vital cellular machines.
Thank you for this incredibly insightful exploration. It's certainly given me a whole new appreciation for these tiny but mighty components. Thanks for listening. And as always, don't forget to like the video and subscribe if you haven't already.